All right, welcome everybody. We are going to shift gears and take it from the human and go back to the plant and talk about what we've seen, what we're going to work on with drying and curing. All right, so to give you a little bit of my background and just an idea of where I come from and where some of this research comes from. I grew up in South Carolina. We're able to grow hemp there, but no cannabis. After grad school, went to San Diego, worked there for about 3.5 years in the medical industry, and then bounced back to my home state, South Carolina, where now we grow hemp. Large-scale production of liners, so clones, lots and lots and lots of other plants. We're vertically integrated, so we do a little bit of everything. But my passion always goes back to the research, and so I'll talk about the research group we formed later. But of course, that's where my passion is, and that's where this data comes from. All right, so. Drying and curing, what is it? I was asking this question back in 2016 when I was in San Diego, and I think the answer then was a lot of I don't know, but I think da 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 da, and I think the answer mostly is still I think, but da 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 da. We've got a few answers. Like the last speaker was saying, one good study just makes you have more questions and more things to figure out. So this is just a bite of where hopefully, will be a long run, lots and lots of answers. Okay, so if you think about a quality smokable flower, because that's what this talk is about, it's not about extract, it's about the flower, no matter where it's grown, harvesting and having it do something else in another situation. But like anything, it's not just about, the second it goes into the dry room. If you want a quality product, you have to start well from the get-go. You need disease and insect-free plants. You need to harvest delicately because you'll knock the trichomes off. And then you go into drying and curing, which is all a part of a path to a high-quality product. And so, why do we care about this? Why do we care about, we grew a beautiful plant, post-harvest. Why does that matter? Well, to some extent, this is for preserving a color. If you dry fast or if you dry improperly, you might turn a really nice green flower to brown. You may handle it roughly, and so you're beating the trichomes off, and so again, the flower may turn brown, maybe for a different reason. You're wanting to preserve the smell. Terpenes, you want to preserve those and the other secondary metabolites that lend to that smell. Talking about smokable flower, you want it to be a smooth smoke. This is one key part that we just do not quite understand. What is it that's happening when you put the flower in a bucket that is going to make it a smooth smoke or not? We don't know. And potency. Potency is not necessarily affected by, as far as cannabinoids go, it's not affected by the drying and curing. But of course, really rough handling, you knock trichomes off, you are in fact reducing potency just because of that mechanical damage. All right, so I come from the ornamental industry, did a lot of work in post-harvest of unrooted cuttings, shipping them all over the United States, and we would look at, how do you ship them properly? What do you need to take away? The ethylene gas. There's gas that builds up. If you think about bananas, you take the ethylene away if you don't want them to ripen. You add ethylene if you want them to ripen. And so, how does that apply to buds, right? Because you're not keeping the plant alive, which you're wanting to do with rooted cuttings. Or if you think about cut flowers, you're not necessarily wanting to save the cut flowers to have roots, but you want to prolong them where they're pretty. And so, research for these sorts of things have been happening for a long, long time. And so, we try to combine the research that's there on those flowers and plants and figure out how can we apply this to cannabis and figure out what's going on in the drying of curing. So that kind of gives us our paths of what we need to explore, but it's not exactly the same. And so a couple words I want you to know and understand going through this talk because they're important. All right, if we think about transpiration, that's basically just the plant losing water. This could be a natural thing. This could be because of it's really hot. It happens at different speeds, but basically, in short, you're thinking of the water coming through the roots, up the plant, which is how a lot of the nutrients are translocated up through the plant, and then out the top. All right, so if you cut the roots off of a plant, if you have a bud, it can still transpire, but it's not going to take up more because it doesn't have roots. All right, and then respiration. So pretty much photosynthesis, right? The plant takes in oxygen and spits out carbon dioxide. I got that backwards. It takes in carbon dioxide and gives us oxygen. But what this is the plant making use of those ions or those photons in the sky and turning it to sugar. So, that's important to remember on some of the next slides. All right, so the harvest part's easy. I'd say it's the least complicated. You can harvest the full plant, you can break off limbs and harvest individual. You can even harvest the buds and dry them individually. I think the biggest takeaway here is, be delicate. If you're in hemp, like you see in this picture, you are not delicate. You do not care how they look. You're just extracting for cannabinoids. But if you are going for smokable flower, not throwing them on top of each other, really removing those trichomes because that's important. And I'll show you on a slide later how important that is. It's something until recently I didn't realize how significant trichome loss is to potency reduction. The other big thing a cultivator would want to think about, and that kind of matters, is leaf removal. So if you have a drying facility that is struggling to keep up as far as reducing humidity, one easy thing you can do is just remove more of the leaves prior to putting it to the dry room. So less physical material to actually have to dry out. Okay, so why do we dry? Of course, number one is this is how we preserve the plant. This is how, think about raisins or coffee. All food products have to have the water removed in order for it not to spoil, to rot. Back in 2016 when I joined the industry, and actually today it is a pretty decent indicator, but when I would ask, well, how do we figure out when dry is dry? What is that factor? And they would say, well, you snap, the stem snap, which, I thought was outrageous at the time, but now it correlates pretty well to the data we've took to back that up. So that's kind of cool to see over time. But the two common methods are water activity, I think is getting more and more popular, which is what the food industry uses, and then moisture content. And those do correlate pretty well together. But water activity is super important because it's not just the water that's there and in the cells, it's the water that those microbes could physically grow on. So once you get down to a certain water activity, you may have spores or whatnot still there present, but they're not going to continue to grow and proliferate. So here's just a quick example. Like I said, other food industries make use of this. It's very important. It's very well known where it needs to be for certain things not to grow. We've got some solid ideas for cannabis. I think more so on how it looks and consumer preference, not necessarily how it correlates with microbial growth. I think that's going to come out where we have a standard of it has to be at this point minimum. Of course, if it's bone dry, we know we're there. But Cannabis is different. It's big and it's got different levels. It's not just a flat piece of dried fruit, let's say. It's complex. So this is a graph I found quite a few years ago, and this is kind of what really got my brain spinning of what's happening in curing specifically. So, if you look at the bottom axis, you've got your water activity, and then you have all the other reactions that are that's happening. This is just in plants in general, at these different water activities. And so, one being straight off the plant, of course, it's wet. It's going to have the capability to feed those microbes. So you have a much higher chance, the higher water activities. As you go down, you have a better chance of not having continual growth of microbes, but you're still getting some of this enzymatic activity that's happening that, we're contributing to this change, this improvement in curing. And then, of course, you have, it doesn't always match what the consumer wants and what science wants, per se. So then you have to include that. All right, well, is this too dry for the consumer? Is this too wet for the consumer? So throwing that into the race as well. And then, of course, once you get to a certain drying point, that enzymatic activity stops. So, this is kind of the big picture of what to investigate, how to investigate, and how it's shaped around water loss, basically. All right, so I made this a few years ago. I've learned a lot since then, but it's still a rough guide, right? So step one, you harvest your plants, and you want to get out water as quickly as possible to stop microbial growth. And it's a tricky balance, because if you're drying fast, it may be harsher conditions, meaning you're losing your terpenes that you want to save. But in order to not have a ruined crop, very fast getting down to that water activity where at least botrytis is not going to grow anymore. Holding on to a higher water activity level for those enzymatic activities to continue, and then, of course, drying it low enough to be at the consumer preference. So this is just a rough guide. There's still tons of work to add to this, but this is what we're looking at for food safety purposes, really. And so, what is drying? It's basically the same thing we do for living plants. We've got our humidity that we want to reach. We've got our temperature that we should reach. But the easier thing to do is to combine those two things, basically, and look at it as the vapor pressure deficit. So, you've got the green, you've got your bud. You think about high humidity. Well, the plant is going to lose the terpenes in the water slower. Makes sense. It's trying to homogenize to its environment. Lower humidity, same thing. But then you throw the variable of temperature into that, it changes it as well. So, I know a few large growers actually are monitoring their dry room by VPD, but I think it definitely will become more and more popular over time. And so just as in case you aren't a cultivator, the norm, what you shoot for is really 60-60. So 60% humidity, 60 Fahrenheit. So pretty cool temperatures. All right, and so this is part from the study we did with the Cannabis Research Coalition, looking at, well, how can we take those two variables as well as adding time and see how long does it take to dry at different temperatures and relative humidity. So we graphed this out, makes sense. Very, very relevant, but this data in a whole allows us to shape a creation of a VPD chart that, if you're a grower, you've probably seen this for your grow room, where there's an area you want to stay in for VPD in order for the plant to lose the moisture or not lose moisture, I guess we could put it. But it's a little different because these don't have roots. You know, we're not growing we're not putting it in a condition to continually grow. We're putting it in a condition to dry the way we want it to. Okay, so obviously, you know, you're getting into really long days of drying because it stayed very moist in the room. Too slow, high risk of botrytis, and then if you're drying it really fast, obviously negative things happen, loss of terpenes, it gets very crumbly, lots of reasons why we need to understand this middle ground to end with a nice product. Okay, and so this is not perfect. There's lots more reps that need to go into making sure this is perfect, but we've got to start somewhere. So this is the VPD chart, looks just like the one that you would use for a grow room. And so we're understanding, you know, how can we really change our humidity or temperature depending on what our facility can achieve to have a certain VPD to lose that moisture. So, you know, this is still in the works, but it's definitely a guide we need as an industry. Okay, I've been working a lot with terpenes the last couple of years, mainly because, you know, I'm in hemp and we play with smokable flower, but terpenes are also very exciting. And so, you know, this is a study that, you know, I first looked at. And, you know, of course you think, well, you're drying. Even in a great condition, you're drying and you're going to lose terpenes. If you're smelling them, they're leaving. Well, how much are you actually losing them? And so in this study, you can see the citation there. In this study, even at really good drying conditions, you know, the best a cultivator could provide, you're still losing over half of your terpenes. It's very significant. And so if I'm a hemp farmer growing for terpenes, this is significant. You know, I have 3 acres of plants and I want to extract terpenes. Well, am I going to go through the steps to dry it, lose half of my terpenes, and then extract the terpenes? Maybe you want to extract them fresh. So this got my mind turning, and we did a few experiments to kind of follow on top of it. All right, so this is, you know, the thought, well, why even dry? And there's different purposes for this, and I'm sure a lot of you have heard, you know, people are going down the road of cryo drying and that sort of thing. But this is a study that was done by Linde, who is a gas company. They sell nitrogen, so they were very interested in doing this. Red Arrow is a marijuana company in Michigan, and then Marcus over at CBDVL. We actually work together down in San Diego. But what this study looked at was, well, with the different ways of drying, how does it change cannabinoids? With different ways of drying, how does it change your terpenes? And so what I just want to make sure that you know, if you're not a scientist, if there's anomalies, ask more questions if you know the author. So for example, if you look at the total cannabinoids, it looks like, okay, well, freeze drying, you know, you're losing a lot. How could that be? Well, when you dig in a little further, what happened in that situation, if you look at the picture on the bottom left, the freeze dryer was pulling way too hard. And so, you know, the tube from the vacuum was just covered in trichomes. There was trichome loss. So it's not that the freeze drying itself was lowering potency. It was a mechanical problem. Next, we look at total terpenes. And so, you know, again, the one that's circled, wow, freeze drying is growing terpenes. But this is, in fact, not true as well, because the fresh flower in this study was not supposed to be dried at the laboratory, and it was. So, you know, it lost some terpenes at the testing facility. So, you know, whereas there's lots of holes, as there is in a lot of research, you take those holes and you fix it for the next experiment, and then you work off that. So that's what we did. All right, so we did an acre's worth. We looked at oven-dried flower. We looked at freeze-dried flower. We looked at flower that was ran through a liquid nitrogen conveyor and then freeze-dried, which saves a lot of time because you're putting in flour that's already froze. And then I had fresh flour tested at the testing facility, and I made sure they did not dry it, which meant I had to do math to account for the moisture, the dilution that they were showing. Not perfect, but we don't have our own testing facility, so this was the best we had. Again, this is for hemp, but I think the big takeaway that we saw with cannabinoids is that, unless you're being mechanically abusive to the plants, all that different drying methods are actually doing is decarboxylating it or not. And so, for hemp, it's not a big deal either way. For marijuana, I guess it's situational. But terpenes is where it got really interesting. So the N2FD, that's nitrogen, and then freeze-dried, oven, and then baseline. So theoretically, the plant, if not manipulated, should have about 2.5% terpenes by dry weight. So that's the bottom number there. Obviously, if you dry it with an oven at harsh temperatures, you're losing a lot. Freeze-dried, it's better, but you're still going to lose something. And then, of course, if you're instantly freezing it right out the field and then freeze-drying it, that was the least lost terpene content. And so that's kind of the high-level look at this experiment, but it gets even more interesting when you look into the individual terpenes. All right, and so this data just came to us like last week, so I haven't ran statistics thoroughly, but I think the takeaways here kind of speak for themselves. So if you look, you've got, a baseline of each terpene by dry weight, which is in that flower. At the bottom, you can see the red indicates the loss of terpenes compared to the baseline. And the green is the gain of terpenes compared to the baseline. And so, what we're looking at here is not just, it was, 2 and 5% differences. I mean, these are significant differences. We're looking at humulene, as it's oven dried, it increased by 84%. I'm not a chemist, but I will definitely be running this across chemists I know and biochemists to help understand what's happening here. There is conversions happening. But yeah, very, very, very interesting. So I think, what the story is here is that no matter how you're drying it, the terpene profile that the plant has as a fresh plant is going to change. So, basically understanding how you want it to smell based off how you dry, you can kind of tweak because you'll end up with something different with each drying technique. Okay, so curing. This is something that for years I questioned. Is something actually happening here, or is it just homogenation? Well, is it removing excess water? Is it homogenizing throughout the container? What is ethylene doing in this process? Are the plants still alive once they've got down to this 0.6 water activity? What's happening with the carbohydrates, the pH, the organic acids? There's so much to question. And once we understand that, then we can optimize and speed things up and be efficient. Okay, so this is an easy one. This is a very easy one. So this was the burp project. If you look at the bottom axis, we have different types of burp. So, once, open the bucket for 30 minutes once a day, no open, an hour for 3 days, et cetera, et cetera. But again, the really big takeaway here is that the bottom portion of the graph, it shows the water activity at the beginning of curing, right? So we've got pretty big ranges of samples. So if you see the, what's this called? The variance. So, pretty big variance of samples water activity. But after it stays in the buckets for 14 days, you go back, you take water activity measurements again, and at first glance, it looks like, well, these flowers are gaining moisture. But that's not actually the case. What's happening is, when you're drying in an environment, the big flowers and the small flowers, everything's drying at different rates. And it comes from the outside of the bud first. So the insides are all different depending on bud size, structure, et cetera. But when you put them into a chamber where they all experience the same humidity, they have time to homogenize throughout the plant, meaning, the flower within the bucket should have all of the same moisture content within. So it's homogenization. That one's very easy to quantify. But then we get to the chemistry side of it, and where do you start? So we look at plants that are similar, but nothing is exactly the same as cannabis. But you've got to start somewhere. So we look at the curing of tobacco. There's lots of ways to cure tobacco, but some of the takeaways here are, well, yeah, it's done for flavor and smokability, but they don't care about terpenes. For tobacco, they're using this process of it kind of being alive, getting rid of the chlorophyll, being in this zombie state to achieve the enzymatic processes they're looking for. But our flowers stay green. So though we may change chlorophyll and harshness, is it actually related to chlorophyll content? It's still green. Other things that are looked at within the tobacco industry are, starch to sugar conversions, degrading carotenes, leaf maturity. All these things have different impact of what the tobacco industry looks as a great product. Again, there's things we can pull from here to test, but it's not a one-for-one. So where do we start? We just want to see what's happening. But we can't do experiments until we have a little idea of what's going on. So we got gas sensors, CO2, O2, and ethylene, and we put them into these sealed jars. All right, so obviously you put a fresh bud into a sealed jar, it's going to mold and rot. It's too wet. But we wanted to understand what's happening with ethylene and what's happening with O2. And so remember back to the definitions. All right, we're going to look at this step by step. The yellow line is humidity, so this is a fresh bud. It's pretty much 100%. Ethylene, this bud is alive. We just picked it. If you're good at rooting, you might even could make this be another plant. It's kind of hard when it's in the flowering stage like that, but in theory, it's still an alive plant. Ethylene goes crazy. These numbers we're seeing of ethylene in this fresh stage, meaning high ethylene in the drying room because it's not dried yet. Ethylene in your grow is super high. And so if you're in a situation in your growing facility where you're not bringing in fresh air because you're wanting to ramp up your CO2, how high are you getting the ethylene in there? I'm not sure. This is something I want to understand. And then is it bad or is it good that it's in there? Or do we want to tweak it right when it's ripening? But takeaway is, in a jar, it gets very high in ethylene. The other takeaway here is, looking at O2 and CO2, this plant is still alive because we see it breathing. We see there is active CO2, there is active O2 being produced. Okay, and so this, on the other hand, is a graph showing CO2 and oxygen after it hits 0.6%. So we've dried it. We're at the end of our dry. It's dead. It's not necessarily breathing anymore. But that does not necessarily mean that there could be other microbes on the plant doing certain things, which we've got some research coming out kind of looking at that, of that interaction. So respiration is insignificant. So that's looking at CO2 and O2, but if we look at ethylene, ethylene is still being produced. So again, why? Not sure. If we manipulate it a certain way, will we get one result versus the other, better or worse? We don't know. But at least now we have a target of what's happening. And so from here we can shape manipulation to see what will make for a better product. And then the question is, well, what does a better product mean? Because then you got to get people involved to give opinions. That's harder than this. Props to the speaker before me. Yeah, so from these very upfront quick experiments to lead into the next ones that will have more significance, what we found is that there's a lot of water. There's a lot of water in the flower, almost 80%. Moist buds have high respiration. So in your dry room where you think it's ambient to maybe outside, it's not. There's a lot of respiration going on. There's a lot of ethylene being produced. But once you get it to, the water activity where you would want to put it into the next stage, the curing, it's not, which was surprising to me. I figured there would still be a lot of gas exchange happening, meaning we burp. And so, is burping actually important? And is there a gas that we're not tracking that is making this, the theory that curing is making for a better smoke? So, as you can see, a lot of, it did this, but what's next? So, going back to this, there's so much more to investigate. It's been a lot of fun, but so the way our research works is the hemp mine, which is my company, and Clemson University, it's where I went to school with my old major professor. We've actually created a coalition, we call it the Cannabis Research Coalition, that looks at, that includes the cannabis industry. It funds the research, and then the research goes to those members. This is stakeholder focused. This is not just research that other researchers want to read. This is research that is going to tell you, all right, well, if you give the plants 2 more hours of light per day, you're going to increase your yield by 20%. And if you're a multi-million-dollar company, that 20% is a big deal. And so, we want to provide research that, yes, is science-based, as you saw, but we want it to be very applicable immediately to a grower. What rooting hormone to use, what's going to take you from having 70% success in rooting to 90%. Things like that will strengthen the industry immediately. Now, of course, a university is involved, meaning they're going to publish, but as you know, not a lot of people are in a hurry to get publications, so it could take a year or 2 to get those out, meaning those members do get that very early perk one-on-one of what they could potentially do to use science to make their grow more efficient, more profitable. And that is it. I don't know if we have any time for questions.
Allison, thank Thank you very much for your work. I appreciate it. As a registered nurse, I did presentation on this for the American Cannabis Nurses Association, but I digress. What is your opinion of the new Bovita packets and those humidity packets that we find so often in our THC products in Colorado? Do you find that that is going to cause issues long term? I just want to hear what you have to say about that. Thank you.
Yeah. So I haven't done a lot of, I can't speak to the microbial effect of it. In theory, you know, there's different choices of humidity packs you can use. So if you're able to stay under that, you know, 0.6 water activity, then in theory, you shouldn't be, you know, introducing water to increase microbial growth. You know, I have seen, with that said, I have seen very dry, crumbly flour, you know, become a better state with these. We are actually working with Bovita. That's where some of the funding comes from. And so, you know, they're looking to make themselves better by having, you know, very nitty-gritty data. So, yeah, I've seen good things, and in theory, it should not have a negative effect for health safety, I guess I could say.
Great, thank you for your work and a great presentation. I'm Will Vaswete from Bayer Scientific, and we do measurements of emission rates in the ambient air, and we see significant emission rates, almost an order of magnitude higher in the drying room than we do in cultivation. Yeah. And so my question is, have you thought about, or have you done anything about using ambient measurements, not the terpenes in the plant, but in the air to try to optimize or look at optimal drying rates? And also, have your alternative drying rates done anything with odor emissions? Those are the two questions.
Oh, goodness. So yes, one of the next steps for this research is looking at the headspace in the curing containers to see, you know, because you open a jar and you smell it, you know, it's coming out, it's volatilizing. And so understanding, you know, what are we losing through this process as far as individual terpenes, and what is being left in the flower, and what makes that difference, and how can it be manipulated. What was your other? Odor. So, you know, to be honest, in hemp, nobody cares about odor. And everywhere I've worked, as far as marijuana goes, that's not been a requirement. So I've not had to go down that road of figuring out how to stop it. I guess I'm just trying to figure out how to keep it. Is the mic on? As far as terpenes, or? We haven't looked at that specifically, but there is a huge difference in drying. You know, if you're pulling off just the stems, you're usually leaving the large bases behind. And so we're definitely able to dry faster because there's less physical water to remove. Anecdotally, not set experimentation, we've not seen much difference as far as cannabinoids or terpenes. So, not exactly.
Great presentation, thank you. Quick question. If you can prevent botrytis in the drying stage, does a longer dry period lead to a better end product? Past like the 15-day point?
Right, I've seen people cure for 6 months, you know, and I've seen people cure for 3 days. And again, by nose,
yeah, for sure.
I've seen the 3-day cure smell just as good as, you know, months and months of cure. But that's just aroma. Is there a longer stage where some of these conversions could happen for maybe a better smoke, which then gets very tough because that's, you know, that's qualitative of how it makes you feel. But at least on data, I've not seen a big difference between the two. And I think with starting to understand this more and more, we eventually will be able to say, okay, well, you're seeing this effect from 3 months, but it's because this is happening over 3 months. But now, since we know what's happening, let's do it in 3 days and achieve the same end result. So that's kind of the goal, is to be able to understand it fully, where we can guide those things. Because, I mean, people aren't curing for 3 months just for fun. I mean, I'm sure they've seen differences over time that are allowing them to make that call. Because if you're a grower, I mean, that's a long time to wait for your money, for your crop.
For sure.
So I imagine there's a reason it's been done traditionally.
Thank you.
Yes, there are actually companies that make these automated devices. I think AutoCure is one, where basically it's a container, and if a humidity gets to a certain amount, or it could be on a timer where it purges. But that's the thing. I have anecdotally seen by smell a great difference between burping and not, but I haven't been able to put a data point behind that yet to match it. So, at this point in time, I can't tell you from science experience that is something you should do. But you should do it, but my science can't tell you why.
Okay, thank you.
Yep.
Hi, thank you. Curing and drying has fueled many conversations in the cultivation community, and we all have our opinions. And I'm one of those guys that believes in a long, slow cure. My flower stays on the stick for over 3 months. And one thing that we've noticed anecdotally among all of us when we break out our bags and our boxes and we start sharing with everybody is we've noticed a difference in plants that are grown full term in full sun retain and have a broader profile, and retain that profile for longer than a flower that's grown indoors from clone that has a forced photo period. And we call it the nutrient density of the flower. Are you taking that into account, or seeing any information with that, and also the curing versus on the stick versus popping the buds off and curing it that way? We notice on the stick we've got longer retention of a broader spectrum, but this is anecdotal.
No, I definitely believe that. If you were to take the same cultivar and put one outdoors and one indoors, so many differences, probably light levels, spectrum of light, all of that's going to affect it. I mean, even, let's say you're growing under HPS lights and you're very close, well, that radiant heat is going to change the terpenes that are there, because you're losing a lot more than you might would under LEDs. And so all of that is changing, to the extent where you can measure that, So, I definitely believe that. And, I would ask you, in your experience, because this matters to apply to the science, why do you choose to do it 3 months?
I was trained by cultivators who have been doing it for generations. And I have 5-year-old flower that I smoke, and it's beautiful, wonderful. You open the bucket and it smells amazing. And we noticed the difference between a triple sec and a Grand Marnier. You get that age with a wine or an alcohol, and you find that it brings out other terpenes. And I know Frenchie Cannoli did some studies with his hash where he's noticing new terpenes being produced, like hashishene. Mm-hmm. And so that aging and that mellowing, we find it's a mellower smoke, and we consider it a connoisseur class, because we notice a lot of the dispensaries, specifically here in Southern California, they won't buy flower that's over 6 months old, and they hate outdoor flower. They want the blingy indoor flash fresh, but we notice if you take a 1-year-old bag of outdoor flower versus a 1-year-old bag of indoor flower, the outdoor still is almost like the day that you were working with the cure, and the indoor doesn't seem to have the longevity.
That's extremely interesting. Again, I can't tell you what or why, but hopefully in a year we'll have some good data points. But what I would like to say is I would love to be able to sample from, batches like this, and us put some data points on, what is being done legacy style, and figure that out. So
Now's the time to do it. There's a whole lot of outdoor flowers sitting unused up north.
yeah.
Wow, okay, yeah, everything he just said also is very much pertinent, but I really appreciate your work so much. It's really helped me over the past couple years understand post-production, so I follow your research. I guess my question would be around ethylene blockers, and if you have experimented with ethylene blockers in post-production.
Yeah, absolutely. So that's something I just squeezed it in 30 minutes, but we did. So in those sealed jars, we would add 1 MCP, so inhibits, and then we actually added ethylene, so adding ethylene. And as far as cannabinoids and terpenes, there was no significant changes, which was extremely surprising. We need to repeat that study. You know, I want to look at it in something bigger than a jar. You know, I think there's some value to that, but I think in curing, it's not as important as it might be in drying and in the grow. You know, we're not seeing those changes in curing, but that doesn't mean that it's not affecting something differently that we hadn't measured yet. So even though there's no change in terpenes or cannabinoids, well, maybe that extra ethylene accumulation or release, because we burp, maybe that is contributing to, for example, starch to sugar conversion. And that's why it's good, but if you're just testing those two simple things, it's gonna look like it doesn't matter. So that is next step. Next step is looking at headspace. It's looking at some of those other factors. So pH, you know, that's a really important one in tobacco, is the pH. You know, that affects the smoke. Looking at total sugars, looking at nutrient content, and pairing that with flushing. Because yes, if you flush, it takes down it takes down the actual nutrient content in the plant, but then what? So.
Thank you. One more little bit is I really do think that it's incredibly important because a lot of people are speaking to how product has to be so fresh in order for it to be sold. And I do feel like we don't have science to back that up. And there's a lot of people speaking publicly that this is a fact and it's not science. So I'd love to see more around that. And thank you again for your work.
Yeah, absolutely. All right, thanks everyone.